Hello, tonight I'm going to be going through some of the senses that we have. The first one I'm going to start with is taste. Uh, there's 5 different tastes that we can have as humans. The first one is sweet, sour, salty, bitter, and umami, or savory. Um, what goes on with the taste buds is we have these things called lingual papillae inside our mouths, and these are the bumps that you see on your tongue. And in between those papillae, we've got our taste buds, which is actually where our receptor cells for taste are going to be at. And those are going to end up tasting things and sending them to the brain. Usually each taste bud is going to have about 100 or 50 to 150 receptor cells. And there's a different receptor cell for a couple of different tastes that we have. So let's just go through those. The salty receptor cell is going to respond to the sodium or potassium ions, and these are going to be dissolved in saliva. So what's going to happen is we have our salty receptor cell. There's going to be an epithelial sodium channel here, and sodium is going to diffuse or enter the cell. This is going to cause a depolarization, which is going to allow for voltage-gated calcium channels to open. Calcium comes into the cell. There's vesicles with neurotransmitter, and the calcium will bind to our snare proteins, and these will be sent out to the neuron, and an action potential will be sent to the brain. Okay, with our sour receptors, it's going to be pretty much the same deal, except it's going to be with the H plus ion going through that um, epithelial sodium channel. So, same thing, H plus is going to enter into the cell, and the calcium voltage gated channel will open. Neuroreceptor will be released onto the neuron. Now, it's a bit different when we do our other tastes. So for sweet, which is activated by glucose or sweet cells, for umami, which is activated by glutamate, and for bitter, which is activated by calcium. These are going to go through a GPCR. So this GPCR is going to come over here. It's going to activate something called PDE1. That's going to send out IP3, which will go to the smooth ER. The smooth ER has a lot of calcium inside, and the IP3 is going to bind to calcium channels. Calcium rushes out into the cell, and the vesicles are able to exit the cell and into the synapse and go to the neuron. So that's pretty much all we have to do with taste. Um, Spicy foods are actually not tasted by our taste receptors, they're by our pain receptors with something called capsaicin, and those are measured in Schofield units. Moving on, smell is similar to taste in that it is a chemoreceptor, chemical receptor. So we draw someone's face and then their nasal passage here. At the top of the nasal passage, right under the skull, there's going to be this thing called the olfactory epithelium. The olfactory epithelium, if we zoom in on it, like I said, it's right underneath our skull, which is this yellow part, bone. And then right above the bone is something called the olfactory bulb. So neurons are going to come down, synapse with another neuron, which is going to go through the little holes in the skull, through our olfactory epithelium, and kind of stick out at the end. At the end, there's going to be a mucus layer, which is created by these epithelial cells. So chemicals from the air that we breathe in are going to be dissolved into this mucus layer, and their chemicals are going to bind onto a GPCR that is in each one of these little ends of the olfactory neurons. So let's blow those up. So here we have our GPCR, a ligand, chemical comes and binds to it. One of these is going to activate something called adenylyl cyclase. Adenylyl cyclase is going to create CAMP, cyclic AMP. Cyclic AMP is going to come over here, and it is going to open up sodium and calcium channels. So those are going to rush into the cell, causing a graded potential. It's going to cause depolarization. That's also going to cause an opening of a chloride channel. That's going to rush out, causing more repolarization. And that will cause an action potential that's sent up to the olfactory bulb and then to the brain. Moving on, we have vision. This is a little more complex, but if we draw out our eye, outside we have our lens, or we have our retina, or our outer lens, and then we have our actual lens in here that is hung up by suspensory ligaments or ciliary ligaments. We have ciliary muscles here that go around in a circle around that lens. And then this is a convex lens, so it's going to bend light rays in towards one point. The closer an object is to the eye, the more the light is going to diverge by the time it hits the lens. So if an object is closer, the ciliary muscles are going to contract. And what this is going to do, so let's draw them all really close together. It's going to cause the lens to bend a little bit because these suspensory ligaments slacken. But if something's further away, the light converges less. So for our lens to be able to focus it on the back of our eye, we're going to need those ciliary muscles to relax. That is going to cause our lens to be more straight. The eye will do three things to focus on an object. Accommodation, which is when the object moves closer, the lens thickens. The eyes will converge, so as an object gets closer, we will start to go kind of cross-eyed to look at it, and the pupils will contract, which increases the depth of focus, allows us to keep objects totally in focus. On the back here, there is something called the pigmented epithelium. And on the pigmented epithelium, there's three cells. There's photoreceptors, something called bipolar cells, and the neurons that are going to come and exit through the optic nerve all at the same spot, which is actually a blind spot in our vision. If we blow this up, draw our pigmented epithelium here, draw our photoreceptor here. What's going to happen is light is going to come in and hit something called rhodopsin. Rhodopsin has something inside of it, we'll blow this up, called cis-11 retinol. When light hits it, it's going to break its double bond and convert it to trans-11 retinol. Doing so is going to stimulate these motor neurons on the G protein to go and stimulate something called PDE, phosphodiester something. PDE is going to come and take cyclic GMP and turn it to just GMP. So it's going to remove that cyclic part, which is going to make it so that ions coming in, such as sodium, potassium, that are depolarizing the cell and allowing for release of neurotransmitter, that is going to be stopped. So light is actually going to inhibit a photoreceptor from releasing our neurotransmitter, which is called glutamate. This is actually good because on the bipolar cell, if it's on center, then the bipolar cell is going to have something called mGluR6 right here. mGluR6. And if mGluR6 is stimulated by the glutamate, it's going to cause the calcium channels here to close. And that makes it so that glutamate cannot be released into our synapse here with a neuron, which has a different receptor called AMPA. Glutamate is going to cause AMPA to open channels and cause an action potential. So basically light is going to inhibit the photoreceptor, which will stimulate the bipolar cell and stimulate the neuron. And that is how phototransduction works. Now, there are things in the eye called receptive fields, and they're kind of like a circle with a circle on the inside. So the inside circle is going to, there's going to be photoreceptors everywhere, all around, and each of them will be affected in the same way by light. They're going to stop making glutamate. But they will affect the bipolar cells in different ways. So if we draw this the other direction, we've got our lateral, our middle, and our lateral cells. Now the lateral cells here are going to, if they're hit by light, they're going to stop making glutamate. But if they aren't hit by light, then the glutamate is going to come out here, and it's going to be changed into something called GABA. So GABA will be increased. GABA is going to come in here, and it's going to inhibit glutamate from coming out of the center cell, which is good, because the less glutamate that comes out here, the more that this bipolar cell will be stimulated if it has an mGluR6 receptor. If it has an AMPA receptor, it will not be stimulated. So if there's light on the middle cell of an on-center receptor and there's not light on the sides, that's going to be how we're going to get maximum action potentials to our brain, and that's actually how we determine the difference between sharp edges and corners. Last but not least, we're going to go through hearing a little bit. So if we draw our ear, the ear canal, got sound waves going down it. And then here at the end, we have our eardrum. It's connected to 3 bones, the ossicles. And those bones, what their job is they're going to amplify any vibrations that come in. So these ones are going to be amplifying their vibrations. And this last little bone here is going to be hooked up to something called the cochlea. At the oval window. Cochlea looks kind of like a snail shell, but if we unroll it a little bit, this is what it kind of looks like. On the top we have the scala vestibuli, and the bottom we have the scala tympani. So scala vestibuli, scala tympani. And in the middle is going to be called the cochlear duct. And basically inside we're going to have this thing called the basilar membrane. It goes all the way from the bottom to the top. The basilar membrane, as these vibrations are entering, they're going to pass across the basilar membrane this entire way. And once they reach a part of the basilar membrane that is the same thickness as the vibrations, it will replicate those same vibrations, and it will cause hair cells to move. So if we just take a cross section of this and we go here, so we have our basilar membrane right there, scalar tympani below, and scalar vestibuli above. And then we're also going to have this thing called the tectorial membrane. So on the top of this basilar membrane, there's going to be these hair cells coming out. And each one of them is going to have part of their hair stuck into the tectorial membrane. So when there's vibrations here in this basilar membrane, the hair cells are going to vibrate back and forth. And each one of these hair cells, it's just blowing up, they're going to have these gated channels on each other. So if this hair cell goes this way, it's going to open up the channel next to it. And that's going to allow for calcium to enter the cell, hyperpolarize, and then it's going to run out below into the scala tympani. And that is what's going to cause action potentials that are going to go to our brain and be interpreted as sound. So yeah, that's about all for this lecture. I'm glad you could join me through this, and thank you very much. Have a good night.